Hi there, this is Rohan Mishra, and in today's topic, we would be discussing on what is virtual screening and what is molecular docking. To begin with, to give you just a link of what we have been doing in our previous lecture, that we have completed topic like target identification and how do we create uh, a protein target for virtual screening or molecular docking. The next topic we dealt with was how do you obtain the compound library. Okay, and once you obtain the compound library, what are the steps that are supposed to be done in order to filter your compound library? Then how do you generate the 3D coordinates of your compounds in your compound library? And finally, how do you uh, prepare your compound library? So now, considering that you have both the things, that is, you know what is the target protein, you have its structure, and you have also prepared a set of compounds for virtual screening. So next step that is needed to be done is that you need to perform this virtual screening, in which you would try to fit the compounds in your compound library into the target protein as such. Okay, we are going to deal with that aspect in today's and next upcoming few videos. Okay, so now that is what I was talking about. When the structure of a therapeutic target is known, or at least if it is available, and this structure of therapeutic target or drug target is usually from X-ray crystallography, or you can develop it using homology modeling if the crystallographic structure is not available, and simultaneously you have set of drugs ready by hand. The most commonly used virtual screening method is molecular docking. So in molecular docking, you do consider some aspects. Okay, so what exactly do you do when we say that I want to perform a molecular docking between these two molecules? I have a protein receptor and I have a ligand, and I want to perform molecular docking. So what exactly do I do? Okay, so if that is the situation, then if you have been given the two molecule, one macromolecule like protein and another one is a small molecule like a small ligand, while you perform molecular docking, we would like to find answers to a few questions. And these questions might be like, do these molecule form a complex? Provided you have a macromolecule and you have a small molecule, will these two molecules form a complex together? Next, you would like to answer the question, if at all it forms the complex, how stable that complex will be? And finally, how will that complex look geometrically? ओके, कि भाई वो कॉम्प्लेक्स तैयार होने के बाद में वो दिखेगा कैसा, और व्हाट पोज will your will your will your ligand take into the protein? Okay, so I would just give you the taste of what exactly do we do in molecular docking as such. Okay, for this I will be using the software called as Python prescription. Of course, we would be dealing with this software in more details in our demonstration section, right? So I would just give you a brief idea about what exactly do you expect when you perform molecular docking, or when you perform virtual screening. So first I will load your receptor molecule first. Okay, I will go to the place where I have placed all of your data files. Okay, so where have I placed it? Okay, so This is your protein molecule, this is your receptor molecule that you have prepared. Now I already have a few results that I would like to show you. They are placed somewhere here. Okay, so now as you can see, what you see in line representation is your protein, and I will zoom in, and what you see in stick representation, that is your ligand. Okay, so this I have already performed this experiment, this molecular docking experiment, in which I have docked this ligand molecule, which is a drug like small molecule, into one of the receptor which has got PDB-ID 2VKU. I have published these results in say some paper which I really don't remember. Anyhow, that is not the matter. What I want you to now follow is that we would try to answer this question. The first question would be, how or will these molecule form a complex. So that is what the complex might look like. This is how your protein, your ligand would go and attach to the given protein. Okay, then I would like to see how stable the complex will be. Now the issue of stability is usually handled by calculating the binding free energy. Okay, and we would calculate the binding free energy for the given set of ligand towards this protein. Okay, and finally you would also like to know how the protein ligand complex would look like. Okay, so here as you can see, you have got at least this protein molecule, and in this protein molecule your ligand can orient in this way. Okay, or it can also take another pose, as you can see over here. Now the ligand took some different pose, right? Or it can take some another pose, another pose, another pose, another pose, another pose, right? So there are different ways in which this ligand can orient itself in the active site of your protein. Okay, so this is what you would expect to do in molecular docking. Okay, so given the protein, given the ligand, okay, how your ligand would orient itself in the active site of that protein. If you want to answer this question, you would proceed for molecular docking. And for each and every pose we have got associated binding free energy. So lower is the binding free energy, that means that particular pose has got greater binding affinity towards the given protein. Okay, so you can see over here, the when the ligand orient itself in this particular pose, the binding free energy for this pose is supposed to be minus 5.87 kilocalorie per mole. So if your ligand acquire the second pose, the binding free energy again decreases to minus 6.34. So that means since the binding free energy of this pose is lower than the previous pose, that means this pose would be more preferred. Why? Because lower the binding free energy, greater is the affinity of this pose towards this ligand. So in this case we would go on calculating the binding free energy that is associated with each and every pose as shown over here. Okay, so this is what you would expect in molecular docking. Given the two molecule, the protein molecule and a ligand molecule, so will they form a complex, how stable that complex can be, and how would that complex geometrically look like. Okay, so now this is how you answer all of these three questions using this molecular docking. Okay, but now let us go to the theoretical details. So now when you say that you model protein ligand interaction, what exactly do you model now? Okay, so say for example, to begin with, a non-covalently bound inhibitor is subject to dynamic equilibrium between the three states. Okay, one of the state is that ligand is in complex with the receptor, which I would call it as R. Okay, and L would correspond to your ligand. Another state can be the uncomplexed state, where both the ligand as well as the receptor, which usually is a protein, are surrounded by solvent molecule. Okay, so if you have a protein ligand system, this system can either be in form of separated protein and ligand, or it can be in form of protein ligand complex. Okay, now one can apply this law of mass action. By applying this law of mass action, one can derive measures of binding free energy from each species. So each species, in the sense, what over here we are talking about two species, protein and ligand. So what is the contribution of the binding free energy of protein and ligand and their complex as such? Okay, so now you have got an equation that reveals the relationship between the binding free energy, that is represented by delta G binding, with the individual concentration of the three species. You can derive the binding free energy if you know the concentration of those species, and the formula is simple: minus RT log of concentration of receptor ligand complex divided by the product of free concentration of receptor and ligand. So in this way, if you know the concentration of receptor ligand complex, concentration of receptor and ligand, and at room temperature, okay, minus RT corresponds to temperature, and this equation would give you estimate of binding free energy. So you can estimate the binding free energy when the concentration of the individual species of the three dynamic equilibrium states are already known. Okay, then the binding free energy can also be described in terms of thermodynamic aspect, okay, as it is composed of enthalpic and entropic contribution. Okay, so this delta G binding can also be figured out using the equation delta H binding, that is delta H minus T delta S. Okay, this is the famous equation. You being chemistry people, you might be already aware of it. So in this equation, the binding entropy delta S is the term derived from the change in degree of order upon complex formation due to change in number of translational, rotational and conformational degrees of freedom. तो delta S क्या correspond करता है यहाँ पर? Delta S corresponds to change in entropy. Okay, what is the disorderness that is created when your protein goes and attaches to your ligand molecule? Okay, so ये delta S का जो binding term है, that is completely dependent on the factors like what is the translational, rotational and conformational degrees of freedom your ligand as well as your protein have. Okay, so that is supposed to be contributing in calculating binding free energy. And the remaining term is delta H, that is change in enthalpy when the molecule goes from uncomplex state to complex state. Okay, so in short, the binding enthalpy, that is this term delta H, sums the change of intra and intermolecular forces. Okay, so what are the forces that exist between these intra and intermolecular forces would eventually contribute the binding free energy in terms of thermodynamic components. Okay, so in this way, delta S as well as delta H is required for calculating binding free energy for a given protein ligand complex. Okay, so normally what happens is that affinity of ligand or drug molecule to its receptor is estimated by computing its binding free energy, as I have already mentioned. Okay, as we know that this binding free energy reflects the binding affinity. Okay, so lower the binding free energy, you can consider that the ligand has got better affinity towards the given target. Okay, now when estimating the binding free energy, one should also keep in mind that the protein and ligand and the complex are usually surrounded by the solvent molecule, which also take parts in the interaction and eventually decides what might be the binding free energy of that complex, right? क्योंकि आपका जो प्रोटीन और ligand है वो हवा में तो रहने वाले हैं नहीं सेल्स में, right? Or even if you perform some in vitro assays, those would be present in some buffer or say some solution, and these factors also do affect the binding free energy values. Okay, so third thing that you should know, in particular water, which is of course a natural solvent for protein, has properties which are difficult to take into account while calculating such binding free energy equations. So we would have to compromise with certain components which are very hard to calculate so far the computations is concerned, राइट तो इसके लिए आपको एक्सपेरिमेंटली वो बाइंडिंग फ्री एनर्जी कैलकुलेट करना पड़ता है. तो अगर आप कोई सॉफ्टवेयर यूज़ कर रहे हैं तो आपको ये थोड़ा ध्यान में रखना पड़ेगा कि मोस्ट ऑफ द सॉफ्टवेयर्स these terms are somehow compromised by either by bypassing these terms or by considering some alternative way. इसके बारे में अपन बाद में बात करने वाले हैं. Okay, so now there are two major aspects of docking program, what we call it as post prediction, and this post prediction is again performed by a specific set of commands, what we call it as confirmer search algorithm or post generation algorithm. And the second aspect of docking program is affinity prediction, और एफिनिटी प्रेडिक्शन का काम किया जाता है स्कोरिंग फंक्शन से. तो आप एक बात फॉर द सेक ऑफ सिंप्लिसिटी समझ के लीजिए कि वॉट एवर डॉकिंग प्रोग्राम यू माइट यूज, okay, doesn't really matter if it is autodoc, if it is gold, if it is hex, if it is glide or discovery studio, whatever docking program, each and every docking program has to perform these two tasks. उसको पोस्ट प्रेडिक्शन करना ही पड़ता है और उसको एफिनिटी प्रेडिक्शन करना ही पड़ता है. तो पोस्ट प्रेडिक्शन में क्या होता है कि यू ट्राई टू प्रेडिक्ट हाउ योर लाइगैंड वुड ओरिएंट इनटू द प्रोटीन्स एक्टिव साइट. Okay, so and this prediction of poses is performed by a specific program in your software called as confirmer search algorithm or pose generation algorithm. And for each and every pose you calculate its affinity. तो ये affinity calculation का काम कौन करता है? Scoring function. Now coming back to our example that I have already shown you. In this case, this is pose number 1. Okay, this is ligand's pose number 1, this is ligand's pose number 1, then you have got pose number 2, you have got pose number 3, you have got pose number 4, so and so forth. So this post prediction का जो काम होता है ये कौन करता है? ये post prediction का काम करता है आपका confirmer search algorithm और post generation algorithm. Okay, and each and every pose is associated with say some binding free energy values, and these binding free energy values reflect the affinity of this pose to your ligand. Okay, so for each pose you need to calculate some affinity value, and this affinity value in case of autodoc is given by binding free energy in terms of kilocalories per mole. So whatever the software it might be, it has to predict the pose, and the prediction of pose work is done by confirmation search algorithm or pose generation algorithm, and the job of affinity prediction is done by scoring function. It is this scoring function that would go and calculate the binding free energy or affinity for each and every pose that is generated by pose prediction algorithm. So this is how these two things work hand in hand. ऐसा नहीं होगा कि आपका सॉफ्टवेयर पहले सारी पोज प्रेडिक्ट करेगा और उसके बाद में हर एक पोज का एफिनिटी निकालेगा. Things don't happen in that way. So these things go hand in hand. Once one pose is predicted, its affinity is calculated, The program then tries to predict another pose, again its affinity is calculated, then it also tries to find out another pose, again its affinity is predicted or calculated. So in this way, this post prediction and affinity prediction goes hand in hand when this computer program works, or when this molecular docking software works as such. Okay, so we are going to deal with what are different conformer search algorithm or post generation algorithm. I would go into details of what are different scoring functions, what is the criteria based on which they calculate the affinity of your ligand towards that protein, what are the things that are considered, ये सारी चीजें अपन ये वाले और आने वाले कुछ लेक्चर में पढ़ने वाले हैं. So in this video, we are going to focus mainly on conformer search algorithms, different type of conformer search algorithm, how the poses are generated as such. And next, or you can say last few lecture, would be based on your scoring function that would give you an idea on how do we calculate the affinity of the given pose towards the receptor as such. Okay, so now this is how you can say is a map of what we are going to deal with in a post prediction. So depending on how the flexibility of the ligand is handled while you generate that complex of your protein and ligand, you can classify different post prediction algorithm. ओके तो इसमें you have three main approaches: rigid body docking approach, flexible ligand docking approach, and flexible ligand and protein approach. Okay, so this flexible ligand approach में you have got three more categories: systematic approach, random और stochastic approach, and simulation approach. तो ये systematic में भी तीन broad categories है. What we call it as conformational search method, then fragmentation method and database method. फ्रेगमेंटेशन मेथड में भी दो टाइप के अल्गोरिदम्स है: प्लेस एंड जॉइन एल्गोरिदम एंड इंक्रीमेंटल कंस्ट्रक्शन एल्गोरिदम. On the other hand, if you go by random or stochastic method for handling the flexibility of ligand, you might have Monte Carlo method, genetic algorithm method, and taboo search method. While if you rely on simulation method, you have MD or molecular dynamics method, as well as pure energy minimization method. So now what I am going to do is that I am going to walk you through with different aspect of each and every method for predicting the poses of given protein ligand complex. Okay, so let us begin our discussion with rigid body docking. So we are over here. Okay, rigid body docking. So in rigid body docking, algorithms have historically, of course, being the first approach. Why? Because these are the easiest way to handle. Using the rigid body docking, it was the earliest created algorithm that were used to screen the set of ligand with respect to their fit to the given target protein. So given the target protein, will the set of ligand fit into my target protein or not? तो ये जो क्वेश्चन है जो पहली बार सॉल्व किया गया था कंप्यूटेशनली, इट वाज on the basis of this rigid body docking algorithms. Okay, so in this rigid body docking methodology, the protein as well as ligand is considered as rigid, right? So we consider that this protein and ligand do not have any kind of flexibility, both are considered as rigid bodies. Okay, so in this rigid body docking protocol, it only allows the translational and rotational degree of freedom. That is, there are only 6 degrees of freedom that are considered while performing rigid body docking. तो ट्रांसलेशनल और रोटेशनल डिग्री ऑफ फ्रीडम है, यानी क्या है कि you are supposed or you are only allowed to only move and rotate the two molecules, okay, when you want to fit them into one another. Okay, so nothing else is allowed. You can either rotate the ligand or you can only translate the ligand. उसको आप move कर सकते हैं और उसको आप rotate कर सकते हैं. Only these two things are considered. Okay, so the softwares that make use of this rigid body docking concept that usually exploit something called as click search based algorithm. Okay, so the earliest software that were developed for rigid body docking were like dock, adam, clicks and hex. Of this, hex is usually still now used for protein docking. Okay, if you want to figure out how two proteins will interact with each other, people still use this hex software. Okay, so say for example dock uses the volume of active site and ligand as a feature to be matched. Okay, so you would calculate the volume of active site and then you would also calculate the volume of ligand, and then try to figure out what portion of your ligand would go and fit into what volume of your active site. Okay, so this is the approach used by doc. On the other hand, adam is the software that is also based on this click search algorithm, and it utilizes the hydrogen bonding as a feature used for matching. Now in adam, well, say for example, what happens is that well, say for example, in the active site of any protein, if you have a hydrogen bond donor, and if there is hydrogen bond acceptor feature in your ligand molecule, then it is considered that this hydrogen bond donor would go and orient itself in the active site where there is hydrogen bond acceptor. Okay, so this matching is usually done in this software called as adam. अभी क्लिक्स क्या एक्सप्लॉइट करता है? इट एक्सप्लॉइट्स डिस्टेंस compatibility of functional groups as the feature to be matched. Okay, so well, say for example, I would like to tell कि okay, if there are two hydrophobic patches in the protein as protein's active site, hydrophobic patch can form if there are hydrophobic amino acid residues like alanine or say valine, isoleucine, right? So these are we'll say for example non-polar amino acid residue. And in the case if there are hydrophobic patches that are present in your ligand as well, well, say for example, if there are methyl group or say alkane groups that can form a hydrophobic patch, then this click kind of tries to match or it tries to figure out what is the minimum distance that these two hydrophobic patches can come together. So now you know that hydrophobic patch attracts another hydrophobic patch, right? So hydrophobic patches attract each other. While if there is, well, say for example, a negative charge in your protein, if the protein's active site contains a aspartate group, aspartate group has got negative charge on it, and well, say for example, your ligand has got nitro group जिसमें आपका negative charge होता है. So this aspartate group cannot really come very close to this nitro group. Okay, so this distance compatibility is usually checked. If such is the case that there is an amino group present in your ligand and a glutamate group present in your protein, well, that means the distance compatibility between these two functional group is high. Why? Because they can come closer to each other. Why? Because they attract each other. So such a kind of distance compatibility of the functional group is checked in the software called as clicks. So it uses the feature to match in terms of distance compatibility of functional groups. On the other hand, the final software hex, it makes use of a similar approach, which is similar to the very conventional fast Fourier transform. Okay, well, let us not go into more details of this fast Fourier transform and its implication, because that would quite go out of the scope of this discussion. Okay, but for time being, you can just remember that Hex makes use of fast Fourier transform for calculating the protein ligand, for predicting these protein ligand complexes as such. Okay, so this is how your rigid body approximation methodology works for finding out the protein ligand complex, how would they orient with respect to each other. Okay, now what are the limitations of this rigid body approximation? One of the major limitation of this rigid body docking algorithm is that it do not somehow take into consideration the conformational flexibility of the ligand. Okay, so what I have told you that when you use this rigid body docking, either only what you can do is that you can only move or displace your ligand, or you can only rotate your ligand as a whole, right? So the thing is that these ligands has got inherent flexibility in the molecule. ओके उसके बॉन्ड के जो मॉलिक्यूल आपका लाइगैंड मॉलिक्यूल होता है उसके अंदर के बॉन्ड में भी फ्लेक्सिबिलिटी होती है. Some bonds are rotatable, and due to rotation of these bonds they can take different conformational states. So this conformational flexibility is not considered when you consider this rigid body approximation. ओके तो लाइगैंड के इंडिविजुअल बॉन्ड्स का रोटेशन इसमें कंसिडर किया नहीं जाता है, right? And so is the case with protein molecule. Both the molecules are considered as rigid, so that the bonds inside the ligand as well as protein will not rotate, and in nature that things don't really happen. Okay, there are flexible bonds in your protein as well as in your ligand, and at physiological condition or at natural condition these molecules would rotate those bond and attain highest conformational flexibility. So well, so for example often a small molecule will have a very large conformational spaces with some of the states can be very low energy state that can form a stable complex with your protein molecule. So these things are not considered when you opt for rigid body approximation or rigid body docking as such. तो लाइगैंड के अंदर के जो इंटरनल बॉन्ड्स होते हैं, the bonds that connect the atoms of your ligand, well, these bonds are supposed to be flexible, and if you rotate those bonds the functional group might rotate and they might take different conformational states with respect to each other, and that thing is somehow not considered in this rigid body approximation. Okay, now let us talk about considering the docking in which you allow the flexibility in ligand, and you won't allow the flexibility in your receptor. ओके तो पहली वाली कैटेगरी में क्या था, रिजिड बॉडी डॉकिंग में वी डोंट कंसिडर फ्लेक्सिबिलिटी इधर इन लाइगैंड और इन रिसेप्टर, दोनों रिजिड है ऐसा अपन ने कंसिडर किया था. But now onwards, in this case, flexible ligand docking, we would consider your protein to be rigid, but we would consider flexibility of your ligand. That we would treat your ligand as flexible rather than rigid. Okay, so based on how do you treat the ligand flexibility, you have got three general categories of how the post prediction algorithm work. Okay, so now if you consider ligand flexibility, what you are doing is that you are allowing to explore all degrees of freedom, that is translational, रोटेशनल एंड वाइब्रेशनल एज वेल. ओके, तो पिछले केस में अपन वाइब्रेशनल डिग्रीज ऑफ फ्रीडम कंसिडर नहीं कर रहे थे. सो नाउ, based on how ligand flexibility is handled, you can divide these software or these algorithm into three main categories: conformational search method, fragmentation method, and database method. So we would go into more details of the concept of what is conformational search method now. Okay, so what happens now in conformational search method, what you do is that you consider only the ligands as flexible. That is, you would allow the rotation of the bonds which are present in your ligand, but you won't allow the rotation of bonds that are present in your protein. Okay, and that too, in conformational search method, what happens is that all of the rotatable bonds in the ligand are systematically rotated through 360 degree. तो अगर कोई बॉन्ड रोटेटेबल है आपके लाइगैंड में, तो उसको आप 360 डिग्री में घुमा सकते हैं, right? Either in one plane by 180 degree and the opposite plane by minus 180 degree. So in all, you can rotate your ligand through 360 degrees. Okay, so now what happens is that when you try to explore by this conformational search method, what you do is that you rotate the bond with a fixed increment for each rotation. Well, say for example, you would first try to figure out ki, okay, in my ligand I have 4 rotatable bonds that can be rotated. So now what you would do is that you would keep you would allow only one of the identified flexible bond to rotate, keeping all the other 3 remaining flexible bond as rigid, and then you would rotate one flexible bond by, well, say for example 30 degree. तो आप क्या करेंगे, 30 डिग्री से हर एक बॉन्ड आप रोटेट करेंगे and try to generate different conformers. Okay, so if you rotate a single bond, it would change its orientation with respect to other functional group that are present in its vicinity. Okay, and then you would repeat until all possible combination have been generated and evaluated, right? So then you would rotate one bond by 30 degrees. I'm just taking the example of 30 degrees. You can decide कि उसको कितने डिग्री से आपको रोटेट करना है. Okay, and in this way you would do the same thing for all the rotatable bonds till you figure out all the possible combination, and for each and every possible combination that you have generated, you would evaluate whether if I have all these combination of rotation, will the ligand properly fit into my protein or not. Okay, so you would repeat this thing unless and until you try out all possible combination of your flexibility as such. Okay, so now such a type of approach is called as a brute force solution. इसे computational language में we call it as brute force solution. You are trying to figure out the brute force solution. तो इसका सबसे बड़ा disadvantage क्या रह सकता है? सबसे बड़ा disadvantage इसका ये है कि the number of structures that are generated increases immensely with the number of rotatable bonds. तो इसमें जितने ज़्यादा rotatable bonds आपके ligand molecule में होंगे, उतना ज़्यादा इसका conformational space बढ़ेगा. The more you would have to test the more conformational space you would have to explore. Okay, and this is usually termed as combinatorial explosion, right? And it is very difficult to implement this concept in its purest form. Why? Because it is quite computationally intensive. हर एक बॉन्ड को रोटेट करके देखो, उसका कन्फर्मर कैलकुलेट करो, and then try to fit it in your protein and try to figure out if it forms a proper complex or not. Okay, so it is quite computationally इंटेंसिव. तो इसका सोल्यूशन ये है कि आप क्या करते हैं, यू ट्राई टू अप्लाई कंस्ट्रेंट्स एंड रिस्ट्रेंट्स ऑन ligand. Okay, so when we say that we are applying constraints, that means that Constraint means when you say that you are applying constraint on say some two atoms, so what exactly you are doing is that you are allowing to a fixed you would allow the bond to rotate at some fixed value. So you won't allow the bond to rotate by plus 180 degree and minus 180 degree. You would constrain them to say only 60 degrees or 40 degrees. Or you can entirely put restraint on say some atoms. Restraint एक बार आपने लगा दिया मतलब क्या कि वो set of atom move ही नहीं होगे. Okay, so this is how you try to reduce the dimensionality of the problem to figure out all the possible rotations and fixed increments as such. तो इससे आपका जो you can say combinatorial explosion का जो problem है वो कुछ हद तक solve हो सकता है. You can somehow reduce the dimensionality of the problem. तो देखो ये constraint लगाना इसलिए ज़रूरी है because we know कि not all bonds in your ligand molecule may be flexible that it can turn by 360 degree in any angle in 360 degree, right? Well, say for example if there is some unsaturation in your compound, तो अगर डबल बॉन्ड रहा तो उसका रोटेशन लिमिटेड रहता है, ट्रिपल बॉन्ड रहा तो उसका रोटेशन और ज़्यादा लिमिटेड हो जाता है, right? So in this way you can apply the constraint based on how that bond is, what is the chemical nature of your bond. So in this way you can either apply constraint or either apply restraint on ligand's flexibility in order to reduce the dimensionality of the problem and thereby solve the problem, solve the problem of finding out the receptor and protein ligand complexes using this conformational search method. And in this conformational search method we have only considered the flexibility of ligand. Mind you that we have not considered the flexibility that is involved in protein. Okay, so the remaining two method I would talk in our next video.